So, what's a human genome? So basically, it's a genomic material. So you have a DNA, and then it's packaged with proteins called histones, and then
Okay.
peptide linked to what we call chromosomes. And these chromosomes are packed into the nucleus of the cell, and we are made up of several trillions of cells. Now, the basic unit are nucleotides A, G, T, and C. So that's basically a human genome. Now, what does it do? It provides instruction. And so instead of this, very simplified. They have the DNA, it's transcribed into RNA, it's like a copy of one strand, and then it's translated into the protein.
Mhm.
So, how is it even translated? So, you have the RNA here, and you have this nucleotides arranged in a particular format. And this ribosome sits on it and they read it. Every three is what I call a codon. And each codon is now code for an amino acid. So that's it here. You now have a string of amino acid, which is a protein. We know this doesn't always happen. In certain situations, the RNA is actually the functional product.
Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think.
So, central dogma, you have DNA, you have the copy RNA, it makes the protein, and it's involved in so many functions, depending on the type of protein in the heart, in the muscle, the eye, etc. Now, what's the role? It's basically a blueprint. It's providing instructions for how we develop, how we function, reproduce, anything. It also explains variation. No two people are the same. Because there are all these variations in our nucleotides and they have slight variants of different proteins, and then we look kind of different. It helps us understand human biology, food, metabolism, even how we respond to how our immune system respond. And then it
Yes.
shows us, basically describes, explains the genetic basis of traits and diseases. Now, there's also evolutionary insights. So it helps us understand how humans even have evolved around the world, evolved and then migrated around the world. So this we're talking about genomics, and the main projects that brought this about were Human Genome Project. So prior to the Human Genome Project, we could sequence, parts of the, genome, genes, short sequences, relatively maybe 1,000 base pairs and whatnot. However, if you pick a haploid cell, we have over 3 billion of those base pairs. Standard sequencing, which was used for the short sequences, wasn't going to cut it. So with the Human Genome Project, they managed to sequence the human genome for the first time. And that was the first time we really understood how many genes we had, which is about 20 to 25,000 base. So it gave us the fundamental information of the human blueprint. And it significantly accelerated study of human biology and then improved practice of medicine. It took
Mm.
it took several years. Uh, Many labs over 13 countries, and the first drug came out in 2003. And for the first time, we actually knew roughly how many genes we had in the genome. And it cost over $2.7 billion to have the first sequence. Right now, it's dropped significantly in the past 20 or so years. And you can sequence a genome for what? A few $100s. Now, what is actually done now is to advance, several technologies, mainly bioinformatics, which is used to analyze all this data because it's a huge amount of data that's generated for sequencing, whole genome sequencing. It's also ushered in the omics era. Now, it's not just about the DNA, we're doing epigenomics. We're looking at transcriptomics, the RNA, we're doing protein, metabolites on a genome-wide scale. So now it's genomic medicine, So this is a new field. And what it does is it uses information from the individual's genome and it helps with the diagnosis, prevention, and treatment. And it's now transforming what we know as traditional healthcare. Because now it's not personalized, it's predictive, and then it's also preventive as well. These are a few applications. You can figure out your risk assessment where once you identify certain genetic variants, it can help you understand your susceptibility to diseases like cancer, cardiovascular disorders, diabetes, etc. And then I mentioned precision medicine earlier. So this helps to tailor your treatment based on your genome. There's gene therapy where you can now go in and, it's a huge area of research right now to correct certain genetic conditions. And it's been demonstrated, in with sickle cell, for example. Now, one area that's really, really interesting is pharmacogenomics. When it comes to medications and all these other compounds we take in, the way it's metabolized is dependent on our genomic makeup. So, if you look at a group of people given the same medication, for example, some might benefit and suffer no toxicity. Some won't benefit and it'll be toxic. Some will benefit and it will also be toxic. Some will benefit and they'll actually know there'll be no toxicity. And it has to do with the variation in genes that metabolize the drug. It is a huge area of interest. Now,
Mm-hmm.
it goes beyond, obvious genetic traits and conditions. Even with infectious diseases, you can have applications here. For instance, there's a genome-wide association study that was done during COVID, the height of COVID, and there were certain areas that, lit up in the genome. For example, the ABO locus, and that has to do with the blood group, the ABO system. And in my
Mm-hmm.
lab, well, we found out that when you group people with COVID according to the blood group, we found out that those who are old were less likely to suffer severe COVID, which was interesting because the majority of Africans actually have blood group O. It wasn't as severe because some of our genetic background actually had an impact on how we're responding to the disease. Now, unfortunately, there are a lot of gaps when it comes to genomic research.
Mm-hmm.
There are a lot of gaps. But why is it important in Africa? So this is what's happening. First of all, the data is really scanty. If you look at this paper from 2019
Mm-hmm.
on GWAS study, you realize that it's heavily Eurocentric. 78% of the data is from European populations, and only 2% are from Africans.
Yes.
Genomic studies are still non-existent. I mean, it's beginning to pick up, but it's really on the low side. So because of the
Mm-hmm.
way you're dealing with, some of the genetic diseases, we don't know what the genetic underpinning is. We don't even know what the environmental risk factors are because they've not been studied in Africa. So, and in addition to that, if you think about traits and genetic diseases, they present with racial and ethnic variations. For example, you pick breast cancer, there's different subtypes. If you look at triple negative breast cancer, for example, it's more common in women from West Africa than
Mm-hmm.
Caucasian. And then if you decide to use just Eurocentric data for Africans, you realize that if you repeat some of the genetic studies, it's not really replicated in some instances. And if you decide you want to resort to African Americans, thinking, well, they're recent migration from Africans, realize they're not fully representative of
Mm-hmm.
Africans. So Africa is really diverse, and they're mostly from West Africa, and they happen to be mixed Native Americans and Caucasians as well. Now you realize that some of these products or resources that are developed on Caucasian backgrounds, when it's just copied and pasted, in Africa, they don't work as well. The favorite drug, somehow it has these variants that are not conducive. And what happens is because it wasn't tested in Africans, once it was used in Africans for treating HIV, it wasn't as effective. So the
Mm-hmm.
whole point, there really is a need for population-based studies because of the differences. Um, Now, I already mentioned that we are underrepresented in, the global genomic database because there's relatively fewer studies that are conducted in black populations.
Yes.
Now, there's so many other ways, there's so many other gaps. And because there are limited studies in these areas, but when it comes to policies and regulations, and there are not a lot of them that are regulating these kinds of studies in our population. There's limited funding and investments in our population, etc. So why is it important for us to conduct genomic studies in Africa? Humans
Mm-hmm.
evolved in Africa over 5 million. 5 millennia after they diverged about 5 million years ago, and then hominids evolved over that period. Now, about 200,000 years ago, that's when smaller populations began to migrate out of Africa, and so the majority of the diversity, the genomic diversity, has remained on the African soil. So because of that, it's the most informative place to discover the genetic basis of complex traits. Because if we evolved, in that environment, we are more likely to identify them. And then when it comes to complex trade, there's a unique contribution of plant populations, and there's this huge opportunity to identify novel loci that are underpinning a lot of genetic traits and condition. Looking at the unique genetic diversity in Africans, there was a study by Lee et al. In 2008. What they did was they looked at microsatellites and SNPs from 50 different populations, unrelated. And basically, they came up with this, figure. Now, as you move away from Africa, you look at the haplotype heterozygous, it drops, which is an indication of how the genetic diversity drops as you get away from Africa. And the whole point is Africa happens to be the most genetically diverse population. Now, in addition to that, which is even more striking, in 2018, Sherman et al. Sequenced about 910 people of African extraction from around the world. And what they did was it was a whole genome sequencing. And then when they finished, they mapped it to the reference genome AG19 at that time. And what they found was that there were extra 10% more bases that were missing from from the reference gene, 10 extra bases. Most of it was intergenic, which can be important as well because it can impact the protein coding regions.
Yeah.
About some of it, let's see, about is it 2000? Oh, I forgot what the number is. Yeah, take that again.
So that's 10% of 10% of the genome, basically.
Yes. So
10% of the genome was different.
Genome was, yes, and most of it were intergenic, but a significant amount of it overlapped with protein coding regions, which indicates that it could have some impact on genes, on coding regions. And this is missing from the reference genome. So that tells you that when it comes to African studies, genomic studies, the yardstick we are using falls short. So that highlights the need for population-based studies.
Yes,
I think
but
I'm going to
10%
skip
is
it.
huge.
Yeah,
It's a huge number.
it is. It
It's a
is.
huge number. They have it like in the paper they mentioned that they found 296.5 megabytes, so 300 megabytes of differences.
Yeah,
That's an amazing.
that is
That's
really
a huge
impressive.
number. That's a
Yes.
huge number.
So
There's
that's a
also
huge gap.
Also, one thing that's important to mention is when you read papers about African genetics, often you have this label African, but it's usually subpopulation. It's not like the whole continent.
Exactly.
Everything is also like these numbers are probably it's underevaluated probably because it's always specific populations inside of Africa. It's not the whole of Africa.
It's just 900 people. So imagine if you had a much bigger, more representative, number. It's going to be huge. It's going to be huge. And so yes, the notion out there is okay. We study a population in Africa and it gives us an idea of the level of diversity on our continent, but it's not. You and I know when you look at figures showing the clustering of diversity, some populations, cluster a bit more tightly. I mean, Asians, etc. But I look at Africans and it's just a spread. There's so much diversity in there. There's so much diversity.
Yes. And also the fact that it's intergenic, most of it is intergenic. It's expected, but it doesn't mean it's not important, because there's a lot of very important things that are in between genes, like functional RNAs, microRNAs, long non-coding RNAs, and
Exactly.
they play a very significant role in the biology.
Yeah, exactly. Gene
Mm.
expression and all of that. So
Yeah,
we
exactly.
definitely
Yeah.
can't rule those parts out.
Yes. So you were going to. I think next slide is about sickle cell disease, right? Yeah. Yeah. Let's talk about sickle cell disease.
Okay. You don't want to talk about cancer.
Let's talk about cancer.
Okay.
Let's do both.
I wish I had some slides. Let's see. No, I don't. Let's
Well, it's
see.
okay if you don't have
Okay.
slides. We can just talk about it.
Okay. So we can talk about, okay, I can talk about how the genetic differences, impact the difference in phenotypes. And for instance, we talk about triple negative breast cancer. And I think I mentioned it earlier, how even in Africa, in Africa, you have, in West Africa, a higher percentage of TNVC compared to East Africa,
Yes.
you know. And then I guess I'm just thinking of how we should take let this flow. Feel like I touched on the phenotypes a little bit when I talked about uh, the TNVC. I think I mentioned it briefly.
Mm-hmm.
Okay, you asked the question.
Yeah. So when you look at cancer,
Mm-hmm.
what specific populations are you looking at?
Okay, so typically I'm looking at blacks and
Yeah. But
Ghanaian
which blacks?
populations as well. West Africa. Okay. Okay. So
Okay. So
okay,
in
let's talk
Ghana.
about
Yeah.
talk about I can say all right. So when it comes to because of differences in the genetic so because of differences in our genetic background,
Yes.
it influences the phenotypes as well. So, like I mentioned earlier, genetic diseases will present through racial and ethnic variation. Now, I study breast cancer, for instance. Now, when you look at breast cancer, it happens to be more aggressive in Black women. It's more
Mm-hmm.
aggressive in West African women and then also African American women. And it so happens that when you can classify breast cancer into subtypes, and the most aggressive subtype is triple negative breast cancer. Now,
Yes.
there's a study that showed, between African Americans and Ghanaians, you have 40, they showed 44 and 49% of TNVC. Now, when they compare this to East Africans, East Africans and Caucasian Europeans, it was between 17 and 24%. Now, when they genotyped, their samples, right, they were able to show that there was a genetic link of TNBC to West Africa. So this basically highlights the fact that even though someone probably lump Africa together, there are differences within the continent. And yeah, sorry, there are differences within the continent. And because of the our unique genetic diversity and of course a higher genetic diversity, there's always the opportunity to identify new novel variants. So I work on breast
Yes.
cancer in Ghanaian women, and and some of the results we saw recently indicated that there were more variants. First of all, we even saw two new BRCA2 mutations, and this was a sample size of 113, and this have never been seen before. So
Mm-hmm.
basically highlights
Yeah.
the reason behind the need to conduct population um, um, based studies. Because, like I mentioned earlier, using resources or products from a
Mm-hmm.
different population, I mean, we are 99.99% identical, but that 0.1% difference is huge, knowing that it's the number of bases. I mean, look at the number of
Yeah.
bases. It's huge.
Yes.
Although we are quite similar, that difference can impact so many things. Like drugs that cause more adverse responses in Africans.
Hmm.
And so that brings me to another study that we did with Yamachi. It was
Yeah.
basically in silico work. And we're looking at, because what the literature shows us when it comes to adverse drug responses, it's more likely to happen in Africans versus other populations. It's because when the drugs are developed in, say, in European populations, they conduct studies, what do you call it, clinical trials, etc. And that's essentially selecting for a particular genetic background. So once these drugs are developed and you just bring it to Africans where it hasn't been tested, there's a lot of variation in the drugs that metabolize the drugs. And because we are more genetically diverse, we tend to have more variations in the, the variants that, that are impacting how these
Hmm.
drugs are metabolized. So what
Yeah.
we saw was that we look at two drugs, and cyclophosphamide and then capecitabine. These are common cancer drugs that are used, for breast cancer and other conditions. So what we saw was that when it came to the deleterious mutations in the genes that are metabolizing this drug, they had a higher frequency in our population, meaning that you are more likely to suffer adverse drug responses when we administer such drug. Once again, that points to the need to conduct population, um, you know, population wide.
Yeah, I mean, there's several things that are super interesting. Like the first one is it's a clear demonstration that we cannot use, and this is something that most people who work on African genetics knew, is that we can't just use African American as a proxy. It doesn't translate to the whole continent. Like,
No, it does not. It does
mm,
not. Because first of all, most African Americans are from West Africa. The majority of them are from West Africa. So people fall back on, okay, it's been a recent migration, okay, a few hundred years, so we should share. We will definitely share some, of course, but it's mostly West African, and so they're not fully representative of us. And then the example I gave with the triple negative breast cancer, for example, it happens to be, the frequency of TNBC was similar between West Africans and African Americans, not with East Africans. So it tells you that it's not fully representative of us, but definitely to some extent West Africa. And beyond that, I mean, with you at the bioinformatician, you have to, when you're dealing with mixed populations, the kind of analysis we have to do, and so it gets a bit complicated. So the thing is, if you're dealing with, you want to understand, the genetic underpinning of conditions in Africans, will be better to use, unadmixed, African populations. Because at the end, the data you get from us will be relevant not just to Africans, but other populations as well, because this is the
yes.
ancestral population. So it'll be more relevant. It'll be relevant to all the other populations. Yeah.
And there's also things like Africa. You look at the Africa is a fascinating continent also, because when you look at the geography of Africa, it has every difficult type of terrain you can imagine. Also,
Yeah.
you have a
Yeah.
forest, you have a desert, you have huge mountains, you have,
Yes.
and that also plays a big role in the genetic diversity because
Yes. Yes.
yeah,
Because when it comes to the genes interact in the environment, so all the what you have in the environment will impact how these genes are expressed. And so a lot of
yes,
times people just focus on genomics without going into epigenomics, which is a huge part of how genes are expressed and how they impact all kinds of diseases. So in different parts of Africa, like you mentioned, they're exposed to different things, but not just the terrain, the geography, I mean, the kind of food, the weather, all of that. And that adds to the complexity. On your last, I think your first podcast on exposomes,
yeah,
in his delivery, what struck me was, okay, I think about exposomes, but the genetic background also has an impact.
yeah. If, it's very complex because exposomics is basically genetics plus what you're exposed to.
Yeah.
And yeah,
Yeah.
and because you can be exposed to so many different things in Africa, and also like just from a population perspective, it has never been easy to move around the whole continent. It
Nope.
has always been hard.
Nope.
I mean, maybe 15,000 years ago it was easy when the Sahara Desert was not a desert. Yeah,
It's
that
not.
also contributes to the genetic diversity, and that explains
Exactly.
why people in East Africa would be very different from people in West Africa because you have to
Exactly.
travel
And
through
the
this.
back, migration and yeah. So if you want to conduct, really understand a genetic, diversity, you're looking at huge numbers. So there's this I forgot what this project is called, but I'm basically looking to sequence the 3 million Africans. Have you seen that?
Hmm.
Yeah, 3 million. Yeah, that's it. And apparently, based on the analysis, that's a number that will fully capture the extent of our diversity. Yeah, sounds expensive, but
Yeah. Also, okay, you mentioned that you found mutation in BRCA, so
yeah,
BRCA. So
yeah,
that's a prime biomarker for breast cancer. So
exactly,
yeah, so
exactly.
that's very very important. It's like one of the top biomarkers that clinicians use to assess what type of cancer it is. So
Yeah,
you basically found three new types
yeah,
that were
yeah,
not known. Yeah,
exactly.
and that also affects the type of therapy that clinicians would give.
Absolutely. And then with the counseling, especially if it's germline, what I described
Mhm.
was in germline. And the interesting thing is, which is not surprising, a lot of the majority of BRCA mutations that have been found are in black patients, and so although it's not surprising to see novel variants, it's reassuring, knowing that okay, that's it can be used for probably precision medicine, for
Mhm.
counseling, etc. Especially the germline is something that can be heritable. And then
Yep.
there's that opportunity to counsel family members and so that they can be more careful or, increase their level of surveillance, to help for early detection. And that being said, because of the fact that the amount of genomic studies is really, really, really small, relatively speaking. I think it contributes to not having, there's not a lot of genetic counseling in our context. Some of these germline variations in cancer causing genes can help with counseling. I think I talked about that. One other issue you have on the continent is a dearth of genetic counselors.
Yes.
So, we know that genetic literacy is a bit low on the side. Compounded with the fact that there's relatively less sequencing on the continent. There's
Hmm.
that. But
Yeah. Also, we can't have we if we don't know the genetic variants of the populations, we can't have precision medicine.
exactly,
So yeah,
exactly. And
and that's
how do you explain that? And another thing too, when you look at
mhm.
the variants that are now being identified in African populations, plus a lot of them are being classified as VUS, variants of unknown significance, because beyond this new, wave of sequencing, a lot of times it stops there. People are associating, oh, this variant with this variant with this, but there's not a lot of functional genomics being, which is another huge gap for Africans. Because if you don't understand the impact of the variants beyond in silico means, you know, how do you use that in drug discovery, in counseling patients, things like that. So that's part of the gap that we are hoping to address on that concept.
That's a big gap. And also when you sequence, there's something the math like the minor allele frequency,
Mm-hmm.
and this is so the thing is if we've probably been discarding a lot of variants because the the frequency is pretty low in other populations, but they might be very high in some African populations,
That's true.
and they haven't been studied. Yeah, which
We are hoping to
another
change
reason.
that. We are
Yeah,
hoping to change that. I mean,
Yeah, we.
other than
I
that,
think.
the
Yeah.
capacity, there are not a lot of geneticists on the continent to begin with, and there's also brain drain.
Mm.
Some will get trained and they
Yeah,
just don't.
brain
They
drain.
just they
Brain
don't come
drain is
back.
real.
They don't come.
Yes. So,
They don't come
some
back.
of us do come back. That's true.
Yes, yes, they do come back. I think I think gradually it's getting there, and I think the rest of the world is becoming aware.
Mhm.
Is becoming aware of the importance of African genomics. Actually, there's a paper that I read where there was this. They were looking at cardiomyopathy, right? And then they found, I think, about five SNPs in linkage disequilibrium that they thought were you know underpinning it in Caucasians. But when they compared this data against an African background, they realized they're not pathogenic. So one of the recommendations from that paper, I'll send it to you, was that
Mhm.
now when you're conducting genomic studies, you probably need to use African sequences as a control, because it'll help you actually really understand the ones that are pathogenic, and important. I mean, after all, of the mother ancestral population, so that's really unique. You want to understand the variants. You want to improve those as well.
Yeah, yeah. I think Africa is definitely like the logistics makes it hard. So there's
Oh yeah,
logistics
absolutely.
is yeah.
I mean, um, funding is an issue uh, when it comes to securing reagent equipment. You
Hmm.
realize it's more expensive for us, which is mind-boggling, because we are considered the poor continent. But
It's more
when you
expensive.
order things like the middlemen, the shipping, the logistics, things on that need to come on
Customs.
ice and all of that. Yes, you need to drop with all of that. So it's really difficult to do research here, but it's changing. I mean, it's
It's
difficult, but
definitely
it's
changing for the
gradually
better.
changing.
Yeah.
Because years
And
ago,
this.
yeah, for instance, the whole exome sequencing we've done in my lab, which I just about to publish, we're the first two to do like a whole exome study in breast cancer, in that population. That was one of the motivation. I mean, that was the motivation for me moving back to Ghana, because I'm doing genetics in the US and I was just keenly aware of the gap in knowledge. So that's what actually motivated me to move back, because I realized, you know, why not be part of the revolution.
Revolution, the genetic
Yeah.
revolution of Africa.
The
Yeah.
genomic revolution.
The genomic revolution. But like honestly, if anyone is doing population genetics, like there's no better place than Africa. It's like you have
Yeah.
for two like human genetics and exposomics. It's the best place to be. Yeah, it's where you have the most variants. It's where you have the biggest exposome. It's where you can make the easiest the easier discoveries are definitely to be made in Africa because you can just sequence anywhere. It's like it's so
The
diverse.
gold mine.
It's a gold mine. It's a gold
Yeah.
mine. You can pick any country, and even like countries have a huge genetic diversity within, and you can use languages as a proxy.
But yeah,
Yeah,
absolutely. Absolutely.
there's a huge diversity. How many languages do you have in Ghana?
Is it like 70 or so? It's quite high. And so
It's
even with
pretty
the
high.
breast cancer samples we collected, samples
Mm,
on Pride. We
mm,
try to go back one generation at least, because
mm,
if you come to Accra, which is a capital, we are
mm.
getting mixed. We're getting that mixed.
Yeah.
My parents are from different tribes, for example. But if you go to the more of the villages, and they're less
Mm,
mixed. But it's
mm,
even looking at, we try to, it wasn't a huge sample size, but even looking
mm,
at some of the groupings, we could see some tribes were more, represented in the data,
mm,
than others. And it's likely though there could be some population, sorry, maybe ethnic, different differences. And we are looking for, if we have a really large enough sample size, it's probably to see something different because
Yeah.
language is an indication of isolation or separation. So if they develop the country, sorry, they have this unique language, it's
Hmm.
likely they've, lived, in isolation
Together.
for a
Yeah.
long time. So then some of
Yes.
these interesting variants will, be, you know, common in some of these tribes. So it'll be interesting to look at that in studies that have really large sample size, and then you can do some meaningful
Yes.
stuff. Yeah. So
That's
we
why I
try we
went.
try to collect those. We definitely try to collect those. It might make
Mhm.
sense of.
Yeah. That's why I think like one of the things that's super important when you're collecting data is metadata,
Yeah,
like the annotations that you put on your samples. And I think if like when data is collected in Africa, I think language should be languages spoken should be part of the metadata
absolutely.
because yeah, because
Absolutely.
that's I think this is the best proxy we have for ethnicity basically in Africa.
Exactly. Yeah, it's
Yeah.
definitely the best proxy. Definitely the
Mm,
best. Yeah, do we have
yeah.
7? It's probably more. And then so imagine the whole of Africa. I really don't know the
Mhm.
number, but probably in the thousand. It's probably in the thousand.
Oh, it's huge. Like I think Benin has like 122 languages.
Oh wow. Yeah. That's a reflection of the the diversity. It is a
It's
reflection
super.
of that.
It's crazy. It's crazy
Yeah.
diverse. I
Yeah.
met like some people from. I met a gentleman from. He's not the only one from West African country, and when you ask them how many languages you speak, they usually don't mention the African languages because just too many. They speak like. So I
I
speak
have a
Spanish, I
friend.
speak French, I speak English. It's like, yeah, nice. But the other ones, oh, there's five
Yeah, they
more.
don't count. Yeah, exactly. I don't know why they don't count. It's a big deal. I have this friend from a guy from my master's, Daniel. He speaks about 10 languages. And he didn't think it was a big deal. I said, this is a huge deal. I don't understand most of those languages. And I'm talking not just dialects. But for example, you can have, there's a language called Akan in Ghana. It has several dialects, maybe 20 dialects. I might be wrong, but I just understand a few of the dialects. It's the same language. But for some of them, I have no idea what they're talking about because it's that diverse. And this guy spoke like 10 languages, some from the north, the south, and he didn't think it was. And I was like, you are a certified polyglot. You can speak so many
Oh
languages. We
man,
don't even
he's more than
appreciate, yeah,
certified.
yeah. You don't appreciate, but it's a lot of language.
Yeah, yeah, and you know, there's also like it's. I think there's all different categories of languages in Africa. You have tonal languages, like you have tonal languages in Ghana. Ghana has tonal
Oh
languages.
yeah, oh, yeah.
Yeah, it's not just Asian languages. Also, West African languages can be tonal. You have there's also I mean, there's the click languages in South
Oh, yeah,
Africa. Like
South
the only languages
Africa.
with clicks. It's like
Yeah.
it's
So it reflects either how old it goes,
yeah,
and then as well as how
e-
how many groups
exactly. It it it
existed.
reflects how
So
deep basically the history, like how old or deep is the history of human beings in Africa and how diverse it is.
yeah,
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 it's it's crazy. Like when you look at a continent like Europe, for example, you have most languages are Indo-European languages,
hmm
so they're basically the same macro family. But Africa is not. It's not the case.
yeah. When I step
That's
out of
not the
Ghana,
case.
I don't speak any other. Even in Ghana, I don't speak a lot of the language. Yeah, quite different.
Yeah,
Quite different.
yeah. So there's also an interesting aspects and like the word like the research is moving now to gene editing and that's something that's to me it's very important because we don't know basically what the variants are and what might be causing some diseases or some phenotypes. We gene editing is not something we can do for most African populations. We wouldn't even know what to edit.
Yes,
So
that's
yeah.
exactly. So my biggest skill set, for my PhD, my first two postdoc, was a lot of functional work, molecular genetics. But then moving back to Ghana, I realized that I had to zoom out again, and that's why I got involved in actually sequencing. Because what we're going to characterize, what kind of functional work we're going to do, you have to now actually sequence. And like we've already, I mean, talked about it, how they're very, in the databases, global databases, there are a lot of, African genomes. So you have to zoom out and do a lot of sequencing before now, once you identify, the causative variants, predicted ones, based on, of course, in silico tools, then can now, be able to characterize them and then make use of them, you know, yeah. So yeah,
So
and then
what
another
did
thing with the genome editing, with the
we,
genome, well, like,
we're not yet there yet. We're the
so
sequencing part, we're the GWAS
yeah,
part.
so we
Like
actually have a
genome-wide
project.
association studies, yeah.
Yeah,
Hmm.
so my master student, her project is to characterize BRCA mutations, not just what we identified, but what were identified in other populations, and we're using CRISPR. So there's a thing with the genome editing. We don't have um, there are not a lot of people in that space, right? Um, but recently, I got involved with, and I did that for when I was in the US. I did that for during my postdocs. But we had this workshop here. We had a grant from International Brain Research Organization, and we had two workshops. And I met this group of people who were involved in CRISPR from Europe, and we recently started a new, initiative to democratize medicine in Africa. It's called Gene for All, and we just launched it. So part of it is to help, train people in Africa. Once we identify the variants, we can go on and train people, and they can be, carrying out this functional work, and so that we can understand how our genome works and how we
Yeah,
can
yeah,
apply it.
that's as you said, we need more people trained in
So
genetics.
we have the first MPhil and
Mm,
PhD programs in West
really?
Africa in medical molecular genetics. So I got
Nice. Like,
accredited this year, and I'm hoping to get you to teach
congratulations.
on the course. Thank you. So these are the West African Genetic Medicine Center at the University of Ghana, and we are going to get you to teach on the course.
Okay.
Bring some AI. So
Okay,
there's
can
a
do
thing too
that.
with AI. This is another thing too with
Mm,
AI that we're also dealing with.
yeah.
I mean, I know I'm preaching to the fire here. That's a space. AI is it's trained on existing data, and if you don't have a lot of African data in there, it's also going to be biased. So it's also now crucial for us to generate enough African data to train AI,
Yes.
So,
Yeah. That's a very important point. Like the number one bias in, I mean, you can simplify and say it's the only bias. Like technically,
mhm.
it's the data. It's yeah, the data set, the distributions that you have in your data set is the biggest bias you can ever find in AI. It's not, yeah, it's not, yeah, yeah. It's true. It's true because if we don't fix that issue, we can't have models that are aware of the African diversity.
Yeah, we'll
So
fix it.
yeah, let's fix it. We're fixing it. We're fixing it. We're working on it actively. Yeah. So
Yeah,
one question I would like to ask you is, there's not that many sequencers in Africa.
no, not too many.
So
Um,
how do you logistically how do you manage that if you need to sequence all over the continent? How do
okay,
you, yeah,
how do we do
yeah,
that?
the logistics of it?
So when I
How do you do that? Yeah.
started, we started sequencing. We started sequencing at the University of Iowa. That's why I schooled in the US. So I was taking my samples there. But subsequently, there's one group at the University of Ghana that had some sequences. At that time, they were focusing more on pathogens, but now they've started sequencing. So I've actually have a set of samples that has been sequenced there. and they're now building upon their, the different equipment, different platforms. And now you have Noguchi has some sequences, and I have private, biotechs like Yamachi also having sequencing. And the cool thing about Yamachi is there's a new initiative that was launched last year or early last year, the Taka project, very early this year. And what it's going to do is to sequence across a number of cancers across Africa. Because then again, if we talk, if you sequence in just one population or in just one country, it's you don't get the spectrum of variants that are predisposing or driving the cancers in Africans, and that's a huge gap. That's a huge area that needs to be filled. So we are sequencing across West East Africa, North probably South Africa as well to give a good snapshot of what's happening. So it's going to be breast, prostate, a number of other cancers. So Yamachi is doing that. Yeah.
Also Central Africa.
I think the central, I know, I can't remember all the countries that. So currently we are gradually forming partnerships in different, different countries. I don't recall seeing Central Africa, but I'm sure it's going to extend to that area. Yeah.
So you basically you leverage existing sequencing capabilities.
So some of them are actually new. Yamachi is just a few years old, and so it's the equipment were acquired specifically for
Okay,
mostly
acquired,
cancer. Yeah, exactly.
Mm-hmm.
But I'll say even before I'm, let's say what 7 8 years ago, there were a lot of sequences in the country. Like I mentioned, we've had some sequences. We're not really doing human stuff. But the past few years, a few other institutions have had it. We have one at WAGMIC here now. We have the next 6550. WAGMIC has quite a number, Noguchi has quite a number now. So we are building, the past few years, we are building on our sequencing capacity. And yeah, it's quite recent. It's quite recent. So because prior to that, you had to ship it elsewhere. I know South Africa was a place to sequence, but I was sequencing in the US. But now the sequencing is being done in house. MRT and all these other places that are doing it in house. Hopefully, I expand. The 13 million genome project. I forgot what it's called. I mean, is it 3 or 13? Sorry, I have to check that. Let me check.
Yeah, it was fine. Okay. Take your time.
See.
Because if you ship, you almost always have a lowering quality of the samples. Like shipment is, shipment is bad for cells.
Yeah, it is. Do you know? I know you probably wouldn't say that, but I carried my samples in a suitcase, but the fresh one I had to put on dry ice, and you can imagine. Okay, so one comes 3 million genomes. So beyond Ghana, I know the other countries that are also acquiring sequences. I know Nigeria has acquired quite a number of sequences. And now that the 3 million project, 3 million sequencing 3 million African project has been launched, it hasn't, I don't know if it started, but I know our sequencing capacity would definitely have to be augmented. Yep. And I know, yes. The interesting thing too, when it comes to, molecular, techniques, I think the Africa experienced a bump during COVID. Mm-hmm. I think Africa experienced a bump during COVID because it was a molecular test, to test for the virus. People were sequencing the virus, here in the University of Ghana. What big Meducci did that. And so I think it was a boost, in some of these areas. Yeah.
Mm-hmm. In training and capabilities.
Yeah, exactly. Yeah. Exactly. And now that there are new programs, the new genetic counseling program I mentioned from, WAGMIC, West African Genetic Medicine Center, started a few years ago. That is also trying to address a gap because now that there's accelerated sequencing, hopefully there'll be a lot of functional work. We understand the variant. You can actually advise, be able to counsel people with some of these, hereditary conditions on our continent. Because, you know, exactly. Some of the previous genetic studies were more targeted. So, for instance, they decided to probably just genotype non variants. But the issue with that is you're looking at non variants in known genes in a diverse population like ours. So you miss out on the novel ones. To expand on that, you need to now conduct whole exome, whole genome, so you see what's not there. But like I said, historically, a lot of the studies here have been targeted, sequencing or genotyping, and these are you're looking at known genes and known variants. But now there's this huge opportunity to identify not just novel genes, but novel variants as well in known genes. Yeah.
Exactly. I think we should move way beyond targeting.
Yeah. And
Yeah,
a lot of people
need.
are getting
Yeah.
interested. That's a good thing. People are appreciating, African genome. Yes, like you're talking about the, logistics, shipping can be really expensive. Sometimes you might get there. What if the dry ice is out? Recently, I purchased some cell lines from the US, and we had a courier ship it. Halfway, they have to stop and replenish the dry ice. And the cells have been in, and so far we've tried to revive several of them. It hasn't worked. And I'm very sure something went wrong along the on the way somewhere. So it can, yeah, shipping, and it's really expensive if you want to ship. Yeah, talking thousands of dollars. So now building, local capacity is the only way to go.
Yeah.
Local capacity,
Yeah.
the samples are here, we sequence, we analyze our data.
It's the only way. And why not start from primary cell lines, African primary cell lines?
Absolutely. So I have a story on that. So part of what
Okay.
I do in my lab is generating cell lines. My PhD students worked on that, and we generated a few primary cell lines, and been trying to immortalize a number of them. Actually, one worked, but there's some contamination, so we had to discard that. So we are still trying to immortalize them. We're using SV40 in human TRT, gene to immortalize them. But we do have some primary cell lines that we've generated that are stored. Yeah, because this was happening again. When you look at the spectrum of cell lines that are used for, all kinds of studies, including drug discovery, they
We know
also
where they come
happen to
from.
be heavily, they happen to be heavily Eurocentric. And if there are differences in, racial, ethnic differences in disease, phenotypes, we are really restricted when we use cells that are mostly, Eurocentric. If you look at prostate cancer, for example, it's more aggressive in Black men. But if you look at the cell lines that are being used for studying prostate cancer, most are from Caucasians. The same with breast. That's the motivation to generate our own cell lines, population relevant cell lines that we can use to study the disease and for other biomedical assays.
Yeah. Yes. I think that would be a huge boost for research in Africa if we can have like population specific cell lines that can be shared around to different
Yeah,
labs.
exactly. I mean, we recognize, like I said earlier, the fact that we are 99.9% identical, but clearly that
Oh,
1%, 0.1% is huge.
yeah.
I thought of
Yeah.
millions of SNPs, for example, and that's huge. I mean, if you look at this, I've been looking at just the phenotype. There are huge differences. So clearly, it's a no-brainer to recognize the fact that if you're dealing with diseases that are underpinned by genetics, there'll be differences as well. So if we could
Yeah.
using less diverse population to develop all these resources to be used by Africans, it's not the best way to go.
Yeah. Yes. And cell lines are cheap also because you can expand them in the lab, which
So yeah,
means Yeah, you don't have to ship them all the time, which
No,
is
indefinitely.
makes a big difference.
Oh yeah, it does. It does. And I
Yeah.
said, when we
Hmm.
buy stuff from the US, from other places, they're even more expensive. I give you an example.
Hmm.
There's a time I needed MCF10 in cell lines. And, I just went on Google to check and automatically links me to the European site. And it's over 900 euros. So I changed I think, what did I do? IP address, something. And then I changed my location and it dropped to 170 USD. And I'm just wondering, why are we being treated, that way? There's a lot of things we are poor, but then I think some of these things are way too expensive. I mean, I know it goes beyond how much the companies are trying want to sell, but they do have middlemen, etc. Because I also recognize that when it comes to the amount of research that is being here, it's
Hmm.
nowhere near what's happening in more developed countries. So there's no motivation, there's no drive to actually establish some of these biotechs here to
Yeah.
produce, reagents and things like that. You can order a reagent that will take months, before it gets here. And you still have to grapple with shipping and the other logistics involved,
Yeah. Yeah.
hoping it
Yeah.
gets here in one piece. If it doesn't work, for example, an antibody, you wait another month, a few months again for it to get back. And if you're in the US, for example, you get an antibody, it doesn't work, the company replaces, you can get it within 24 hours, a couple of days, and you can, so yeah, it's not easy doing research here. But I think
Yes.
now that the research ecosystem is expanding, perhaps to get to a point where we reach the critical point where some of these biotechs would recognize the need and um, accept that it will be economically viable to set up over here, and do that. And I know, yeah, other than like I said, some of the companies will probably be selling at a regular price, but then we have to deal with the shipping. Not just that, taxes.
Taxes.
Yeah, but we think, yeah, but I think we'll work through that. We're working through it because we don't have a choice, do we?
Hmm.
It will
I
benefit
mean,
everyone.
someone has to do it.
Yes, it will benefit everyone at the
Someone has
end
to
of the
do
day.
it. Yeah.
Yeah.
I like talking about taxes.
I have your own set of tax issues.
Yeah, it's true. Actually, I actually enjoy talking about taxes because I think like most of like a lot of things can be explained by taxation. It's like
Hmm.
things that happen, also things that don't happen. Can be explained through a careful analysis of the taxation
Yeah.
system.
Mhm.
But it's true. Like there's an issue. There's the economy of scale because when you're in the US, like North America, you have a lot of universities, so you can,
Yeah.
as a company, you can make there's a scale that allows you to lower the prices.
Hmm.
However, if you send it to Africa, there's maybe one lab or two that
Hmm.
might need your services. There's also the exchange rate, the
Oh yeah.
currencies in Africa. Yeah, that's
Yeah. Yeah. The inflation and
yeah.
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 So all that makes it it's not let's say more challenging, more interesting to deal with.
Yeah. Challenging and interesting.
Yeah. It's but it gives you an interesting set of skills. And
You become a
forces you to think
magician, not
outside
just
of
a
the box.
scientist. Not
Mm.
just a scientist. You become a magician as well.
Yes, yes, you become a magician. But it's but the kind of things that you're doing in Ghana today, I think it would have been hard to imagine it like even 10 years ago.
Oh yeah, absolutely. Absolutely. And I remember 8 years ago, wasn't it? I was just saying that 8 years ago. And I got a first cell line in the department. I guess Hextel. And I remember I couldn't get, I ordered stuff and I just couldn't even get trypsin in the whole building. And I had to come to Noguchi. I got a small tube of trypsin and then I had to figure out what I was going to do next. But funny enough, by next morning the cells were contaminated. I've worked with cell lines for 10 years. I mean, in Iowa, like I've done anything you can imagine with the CRISPR, with the regulatable cell lines. I've done, all of that. I never had any contamination. No, so this was the first time. The next morning, the cells were black. I mean, they were still thriving, but they were black. I didn't know what they were. But yes, but things have changed now. There are a lot of cell lines there. We managed to
Mm-hmm.
build a good, catalog of it. But yeah, things now looking back, there are a lot of sequences. We have a conducive environment. It's things that have you know changed quite quickly.
Yeah.
A
Yeah. Yeah.
lot.
Yeah.
And all because
Yeah.
of funding. Because of funding, some funding that came,
Mm-hmm. Yeah.
World Bank
And because
funding, other
it's not just funding,
projects.
it's also brains coming back.
I guess so.
Just like,
But the brain is horrendous. The brain drain is
yeah.
horrendous.
Mm.
Honestly, even when I moved to the US, I didn't think I was moving back anytime soon. I knew I was going to move. But then I was just thinking, at that point, Agbip had a World Bank, um, project. They had a World Bank project grant. And so that's when they started buying the sequences. They had this, I mean, the flow, all of that. And so then I had a mix from way back, went to college together. And she was the one who I started talking to about it. And I realized, okay, I I was reluctant to move back immediately because I felt with what I was used to, the kind of cutting edge science I was doing my CRISPR and all of that. I felt I couldn't do that in Ghana. But then gradually, I also thought that, you know what, there's a huge gap when it comes to genetics. Do I have to, why don't I be part of the story, And then thirdly, I said, okay, our group is building, a conducive research environment. And at the back of my head, I'm like, okay, if there's something I can do here, I can always collaborate with, other researchers from more, established university like my old university. So that's my motivation for moving because I realized the research environment was becoming, more conducive for, the kind of research that I could do. And that's partly why I was able to move back. And things have changed significantly. And I know 15 years ago, the department probably was still doing, the basic backend test, the usual color change and all of, um, chromatography and all of that. But now there's been a huge change. And it's not just at the University of Ghana, the other universities that are, expanding and all of that. And then there's expansion across Africa. I just
Yes.
hope, uh, our respective government recognize, the importance of conducting such research. We can't always rely on resources from other places because
Mm.
drugs will come and they won't work for most people, they'll work for some people, and the address drug responses. Now that we're in the precision medicine era, it's crucial that we get on board. And the only way we
Yes.
can do that is to have population, based studies so that our data will be
Yeah.
included. Of course, AI only has been,
Yeah.
so
And
yeah.
this, I don't think there's an option because if you don't invest in research, you don't get precision medicine. You say no to precision medicine. It's like, no, we don't need it.
So no, we don't like it. You guys can have it.
So you no precision medicine and drugs that don't work as well that you have to pay.
Yeah.
You have to buy them still.
And so the interesting thing is, we have a lot of resources. When you talk about, local plants, that's another big area that's, kind of flourishing now. A lot of these really good drugs have their roots, in quotes, in, traditional medicine. And you have a huge, um, catalog of such plants. And so now with the kind of capacity we are building, we are going beyond, okay, is this clean this cells? It's just beyond that and do actual molecular work. So, I mean, I've had some collaborations with other departments, chemistry and pharmacy, where we've done some, work in my lab, actually looking at, gene expression, not just whether the cells are dying or not, And subsequently, if we're able to develop, generate our own cell lines, more population, relevant models, we'll be able to, I think we'll be able to, at least discover a lot of drugs, because there are some that are being used traditionally, which, I mean, they claim to cure this and cure this and cure this. But, as a scientist, of course, we need to test this rigorously, rigorously.
Yes.
I know there are studies being done, in maybe rats, for example, looking at toxicity and all of that. But I
Mhm.
think once we generate more population relevant models, we have a good shot at, not just waiting for others to, we know it's expensive. You can't wait for other people to develop drugs. They probably wouldn't even test in, all populations anyways than people. But I think we have that opportunity also, develop our own drugs. And yeah.
Yes. Yeah, we can. I mean, the technology is there, and the hardest part is, and this is interesting. I mean, it's happening in Ghana. It's also happening in Morocco, so I can see it like firsthand. But there's a very hard part that you you don't get trained to do that as a researcher, and that's building the whole infrastructure from the ground up.
Yeah, more than a scientist.
What the scientist, you need to
Yeah.
build the thing.
Yeah.
Yeah,
Yeah.
and like it's not easy. You need to. I mean, if we talk just about, you need the instruments. You need to know what type of instruments you need. You need to have
Yeah.
vision, because you don't need to buy. You don't want to buy obsolete instruments, or instruments that will become obsolete very quickly. You need to train the people to use them.
Oh, Akosombo, yeah. And my dad is from Bako. Yeah. Small world.
Right?
Uncle. All right.
You can invite her.
It's such a small world. Sorry about that. I think she's so excited. There's a training in the building, right? We have a brand new building, and we
You
rent it
told
out
me.
for activity. Yeah, so I think she's here for that. So I think she's just close, and I think she's just excited. She saw a family member.
She's happy to see you. She's like, oh,
Yeah,
I found a lost relative. She- yeah.
yeah. So
So
okay.
let me know when you need to leave. We can, if you need to, just let me know, and we can stop anytime. But yeah, so I was saying, the thing that you don't get trained to do is building the whole infrastructure from the ground up. So getting the instruments, getting the right type of instruments,
Mm-hmm.
getting them so they don't become obsolete in like 1 or 2 years.
Mm-hmm. Mm-hmm.
So you need vision for the type of research that you want to do, where the research is going. You need to stay competitive. You need to do it in a very cost effective way, and you need to train the people
Yeah.
to use them. So that's
Yeah.
that's a lot. That's a lot of work.
I feel like being a scientist, it's you should have that interest. Imagine just pipetting the same stuff over and over again, doing the experiments over and over again. It's not working, but you're just driven by your curiosity to figure out what's happening. It's it has it's the drive. There's this drive we need to have being a scientist. If not,
Yeah,
you wouldn't make it for the science. You're
exactly.
absolutely right.
Like,
It's not the motivation isn't necessarily much. That curiosity, that's excitement. Oh, you find this and you're excited, you know,
motivation is
excited.
not money, and
Yeah. Like I
this is
said, in
why.
Braca mutation. Oh, this is nice. This is nice. It's just like, how is that going to feed me?
But this is really nice. It's super important, especially like
Yeah.
I've heard clinicians talks about that, but it was anecdotal. They would say, "Oh, I've seen these patients. Like I've seen some patients in my life. I followed like the what I was supposed to do." Given the
Mm-hmm.
BRCA mutation that I've seen, like BRCA was mutated. Usually, don't even know what mutation it is. You just have like mutated BRCA. That's it.
Yeah, that's it.
They, that's it. That's it. They will give treatment non-responsive.
Yeah.
Yeah,
Yeah.
because it's a different mutation. It's a different type of
Exactly. That's
yeah,
exactly. And as a
functionally it's different.
scientist, you just get excited. What's happening? What is happening? So back to this studies. Sometimes some of the genetic conditions they'll say, "Oh, it's not been seen in Africa," and all of that. So one of them is cystic fibrosis. I worked on cystic fibrosis for my first postdoc, and I mean, the literature will talk about okay, it's less common in black people. It's more common in white, and it's quite common in white. I think it's one of the genes they look out for the newborn screening, TSCR mutation, because of how common, but not as common as sickle cell. Anyway, so the literature always talks about they haven't seen it in Africa, you know. But at the back of my head, I'm like, okay, I know there's some conditions that are probably more common in certain populations. Sickle cell being one of them. But in my head, the CFTR gene has about like 2,000 mutations. How is it that we don't have it? And a couple of years ago, someone sent me a paper. Two children had sickle cell in Ghana. Sorry, two children had cystic fibrosis in Ghana. So it's the same thing because we've not done extensive research when it comes to the epidemiology of a lot of these genetic conditions. They say, oh, it's
Mm.
low in Africa, which is possible, but it's absent in Africa. But it might be there, but we just don't see it because some of these
Yeah.
conditions might be lethal, early on in life. But if the means for even diagnosing and identify is not there, you will just not think it's there. And now a lot of the genetic conditions, like the obvious ones, the doctors rely
Mm.
on phenotype. They
Yes.
rely on phenotypes. But then there's this opportunity to see, because for some of them, they just can't phenotype it. Based on the literature, they usually follow maybe a particular, what you call, to identify
Hmm.
some and guess, okay, this is what it is. But there are some of them, they just can't match to anything. And the only way you can do that is to sequence. Especially if the suspicion is that it's a genetic condition. You wouldn't know until you do that.
Hmm. Yeah.
But we
That's
will
it.
get
Me
there.
too. We will get there. I don't believe it necessary when people say it's not present in Africa because at the
Mhm.
back of my head there's always maybe you haven't sequenced the right Africans. Like
Exactly.
yeah, for
Exactly.
a very long time the only like workable genetic information that we had were from Yoruba people in Nigeria. That's it.
Oh,
It's like
yeah. Just
that's
one
that was
population. Yeah.
exactly. It was
Yeah.
the only one that was big enough to do anything
Yeah.
with it.
Yeah.
It's non-representative for a lot of different populations in Africa. Now that's
Yeah.
and yeah, yes, and this is also why I really want to have this discussion with you. It's because I want people to understand that it's Africa is extremely diverse genetically, and usually all this genetic diversity is lumped into one single label.
But
Africans should know about that because they should know that the diversity, the fact that Africa is extremely diverse has real serious implications when it comes
absolutely.
when it comes to health, when it comes to precision medicine, when it comes to the novel technologies of gene editing, when it comes to like if we don't sequence uh, like now we're talking about helping people cure genetic diseases. Like this is the level we're at the moment. Africa is if we don't sequence, Africa can never get on that boat.
No, we cannot. Cannot. Yeah, I'd like to add that. Yeah, we're talking about sequencing a lot, and I briefly touched on,
Yeah.
transcriptomics. We should try not to leave out epigenetics. It's a huge
Yes.
part. Yes, huge
Epigenetics.
part. I realize most people are just interested, oh, let's sequence, let's sequence. Yeah,
Yes.
epigenetics is massive. We can't do without it.
Wow.
So if you want to get on that boost, that precision medicine boost, we definitely
Yes.
should get on board. I mean, people they're developing drugs and targeting microRNAs and all of that, trying to change the chromatin, state and all of that. Some of these new cancer drugs. and we really don't fully understand what's happening here. So beyond the genomics, which need the transcriptomics, all the other omics. Now, thanks to the Human Genome Project, it's actually in this era of omics. We should take full advantage of that. All of it. So the funny thing is, even though I'm in the genetic space and I really understand, I can appreciate the diversity. I was so surprised when I found out that triple negative breast cancer was less prevalent in East African. I know I've mentioned it already, but I found it really fascinating. And I remember the first time I saw it in the literature, I was thinking, oh, maybe sometimes it wasn't quite accurate until that paper from just a couple of years ago, where they actually did the genotyping and they could see, you know, that is if the East Africans were comparable to Caucasians, and then the West Africans were comparable to the African Americans. I really doubted even with that, but yeah, we can all now appreciate.
Yes, me too. You told me about that paper. I was
Yeah,
surprised. But
yeah,
it's always the case with each time I look at a paper that's well done on African genetics, there's always a big surprise. Like
yeah.
always.
And I found the Moroccan one to really fascinating.
Yes,
Really fascinating.
the E1,
How did
B1,
that
B1.
how East African men end up there? And
Marrying European women.
yes. You see. So
No.
I mean, it's telling us so much about human history.
Yes.
Not just now we talk about precision medicine and all, but it's telling us so much about history and the migration. Who
Mm-hmm.
would have thought? Who would have thought?
Probably something very, very old because there's like 15, yeah, probably like 15,000 years ago, which is not that far genetically. Like the
Mm-hmm.
Sahara was not a desert, so people are probably moving from one
Yeah, it
place
was
to the other, from
easier.
East Africa. But
Mm-hmm.
the whole of the Sahara was basically a savanna, so you could just walk the whole thing. And there were probably rivers also that they could navigate. So maybe
Yeah,
people were moving around
much
in
easier than
yeah, much easier
now.
than they do than now. Yeah, that
Yeah,
could explain it. I don't know. That's just my theory, but because it's also it's an interesting haplotype.
it is. It is. It is. I'm just curious about
Mm.
what the findings once they manage to you know give us a good snapshot of the diversity across Africa. It's, I'm just looking forward to what kind of stories it's going to tell, what kind of answers it's going to provide. And
Yes.
imagine, we have able to have a good representation of the worldwide genomic you know um, diversity. Can you imagine? I think give a clearer picture of migration. Even now, we have a good idea of how humans migrated, and I think even some of that were determined before the advent of genomics. So I can imagine how these paths and the migration patterns will be refined once you have a good snapshot of worldwide genomic diversity. That'll be. I'm looking forward to it. I sound so much like a scientist right now. That's what I'm excited to see.
Exciting about this. Exciting
I'm
about this kind
excited
of
about this.
questions. You were talking about this, and I was thinking about Europe
Mm-hmm.
because I think like there's two things that are fascinating because they would use languages and culture basically to map out before we had genetics to map
Mm-hmm.
out people's migration. And I
Mm-hmm.
think I think Finnish and Hungarian are related,
Oh,
which is yeah, it's very very interesting. And then you have the Basque language that's related to nothing around it. It's just like they just there we don't know where they came from. Um,
yeah.
and there's these two situations are there's probably a lot of them, a lot of these situations in Africa, like pockets of
Oh, yeah.
populations
Yeah.
that yeah,
Yeah. I'm
some
I'm just I'm just
migrations.
curious
Mm,
to see. I'm excited to see what it tells
yeah,
us.
yeah. It's like I think it probably happened. Like I don't know, like something happened in the past, and then some catastrophe, and then people moved around, and one pocket got somewhere in
Mm-hmm.
Ghana, and the other one stayed in Central Africa, but they share some very strong genetic
Yeah. So there's this
ancestry.
story. This there's I don't know, we learned that in cultural studies in school where we believe one of the tribes. So the tribe that is from the indigenous tribes for Accra. They're called the
Mm-hmm.
Gans, and it's believed they're from Nigeria. I mean, it's in our history books, and I think this would be something interesting to to study it, to kind of compare, the genetics, people in Accra, the Ga people, and then, people in where they presume they migrated from. It's something we learned in history. Everybody knows that, oh, gas, what else? Just get about, I don't know how far back that goes. I'll have to look it up. But it's believed historically that they migrated all the way from Nigeria to Ghana. So that's something that can be resolved looking at their genetics, right? Yeah. The prospects are endless, not just disease. But well, move and all of that,
Yeah,
you know.
and there's a lot of cultural things like that, like stories that people had been telling for a very long time.
Yeah.
Yeah, and this can get very controversial, so I'll start. I'll get to a non-controversial one. So one that's not that will not get anyone excited or too much
Too
excited
excited.
too much about what I'm going to say. But the Romans. I don't think there are any more Romans, like true Romans left living, but they their
Mhm.
story was they come from Troy. Maybe there were some
Okay.
people who came from that region, but definitely not all the Romans.
Mm, interesting.
Examples like that where people would just like
Yeah.
latch on for cultural reasons or historical reasons to
Yeah.
some other population in some other region and then say we come from there, but they actually don't. Like genetically, when you look at
That's
it, it's
true.
people don't realize how easy it is to change the color of the skin of your offspring. It doesn't take that many generations,
Exactly. Exactly.
like two, three.
Exactly.
That's it, max.
You're done. You're done.
That's not hard
But
to do.
20,
Yeah.
23andMe has been really eye opening, yeah, for a
Mm.
lot of people.
I have a Chinese friend who's like 1 or 2% North African.
Humans have been very busy, really, really
Yeah.
busy. When someone ends up, and
I
yeah,
mean, we've been traveling, like we've been walking
uh,
around, we've been taking boats, we've been doing a lot of stuff. So,
yeah. It's,
yeah.
it's it's fascinating how some of them you just in our
Mm.
current state, we can't imagine how people got that far. And I'm like, how did they even get there?